Welcome to problem set 23. Our problems today are from the topic proteins. Before we get started, just a quick reminder that the worksheet to accompany this problem set video is available online through my website. And with that said, let's get started with number 1. Given that the side group of alanine is CH3 and the side group of glycine is hydrogen, write out a reaction to show these two amino acids forming the dipeptide alanyl glycine, and then complete A through C below. All right, so you need to be able to draw the general structure of the amino acid on your own, and then the side groups are given in the problem. So, starting out with the amine, and then the alpha carbon, which is right next to the carboxylic acid. We are given that the side group for alanine is a CH3, so I'm going to fill that in here. And I'll abbreviate alanine with the 3-letter abbreviation ALA. And we're going to show it reacting with glycine. So same thing, except now the side group is a hydrogen. So remember to change that. So an amine, the alpha carbon, which has a side group that is a hydrogen, and the carboxylic acid. Okay, and if it helps you, you can use the arrow to help you remember the new bond that's going to form here between the nitrogen and the carbon of the carbonyl. You're going to lose the OH from the carboxylic acid and one of the hydrogens from the amine, which is why your byproduct is H2O. Okay, so I should label this glycine. GLY is a 3-letter abbreviation, and my new dipeptide will look like this. Okay, so notice I just copied all this down. None of those bonds changed. Now I'm to the new bond between the carbonyl and the nitrogen, which is my peptide bond. And then the rest of this molecule is unchanged, so I just copy it over here. Don't forget your byproduct plus H2O. Okay, so this is, oops, not glycol. This is alanyl glycine. And again, I'll use the 3-letter abbreviations for that. Okay, so now we're going to answer A through C for number 1. A, label the N-Terminus and the C-terminus of the dipeptide. So let's do that in a different color. Let's go to blue. The N-terminus is the side that has the free amine. And remember that that is traditionally going to be drawn on the left side of the molecule. And the C-terminus is the side of this dipeptide that has the free carboxylic acid. And that's going to traditionally be drawn on the right side. So here is our C-terminus. Okay, so for part A, I'll just write C above, because I labeled it up there in the drawing. Okay, part B, label the peptide bond. So remember, the peptide bond is the amide bond where the carboxylic acid and the amine came together and formed this new bond here. Okay, so for part B, again, I'll write C above because I labeled it up there in our drawing. And last but not least for number 1, part C. If the 2 amino acids were switched to make glycosalanine, would the new dipeptide be identical to the one you've drawn above? Explain. So if we switched the order of the amino acids, so we had glycine first, so glycosalanine, would they be the same? The answer is no. Glycosalanine is not the same as alanyl glycine. And the reason is because in alanyl glycine, the N-Terminus is off of the alanine unit, and the C-terminus is off of the glycine unit. If you switch the order, then the N-terminus will be off of the glycine unit, and the C-terminus will be off of the alanine unit. So similar, they're made up of the same 2 amino acids, but they are not the same peptides. So let's write here. When the order is switched, the glycine unit will have the free amine that is at the N-terminus, and the alanine will have the free carboxylic acid. At the C-terminus. And that A doesn't have to be capitalized. Since I'm not using the abbreviation, I won't capitalize that. Okay, so similar, made up of the same 2 amino acids, but not identical dipeptides. All right, let's go on to number 2. Okay, number 2. Which of the following side groups would be expected to participate in hydrophobic interactions? Circle all that apply. So I've got an A through D up here. They're drawn on the worksheet, written out on the worksheet, and then I've drawn them out on the board as well. Let's go to the blue pen. So we're asking which ones would participate in hydrophobic interactions. In other words, which of these side groups are non-polar? Which ones are non-polar? So we can see that A contains just carbon-carbon and carbon-hydrogen bonds. Carbon-carbon bonds and carbon-hydrogen bonds are non-polar. So we would expect A to participate in hydrophobic interactions. It is a non-polar neutral side group. B, same thing. We have all carbon-carbon and carbon-hydrogen bonds. So let's circle B. C has an OH, which is a very polar side group. Makes this a very polar neutral side group. So do not circle C. C would not participate in hydrophobic interactions. C is a very hydrophilic side group. And D is a charged side group. It's a basic side group. Remember, at physiological pH, those will be protonated and therefore charged. And so we wouldn't expect D to participate in hydrophobic interactions. It's kind of like those charged phosphate groups that we saw on our phospholipids. The phosphate unit was very hydrophilic. Same thing here. This charged unit would be therefore hydrophilic, not hydrophobic. So just A and B for number 2. Okay, number 3. The 2 separate protein chains of insulin are held together by disulfide bonds. This is a specific example of the blank structure of the overall protein. So is it A, an example of the primary structure, B, an example of the secondary structure, C, an example of the tertiary structure, or D, an example of the quaternary structure? This is an example of D, the quaternary structure. Because remember that the quaternary structure refers to the way in which two or more polypeptide chains will come together to make up an overall functioning protein. Not all proteins have the quaternary structure because not all proteins are composed of two or more separate chains of amino acids. In this case, we're talking about the way two separate chains are held together by disulfide bonds, the quaternary structure. All right, let's see, we're running out of room, so let's go ahead and erase here before we go on to number 4. Okay, number 4. Which of the following amino acids would you expect to find on the outside, and which would you expect to find on the inside of a globular protein? So this is a protein that needs to interact with the aqueous environment of the body. So we would expect any hydrophilic side chains to be oriented toward the outside of the globular protein, and any hydrophobic side chains to be coagulating away from the aqueous environment toward the inside of the globular protein. And I've given you A through D. In the problem, I only wrote the name, but you'll see that up here on the board, I went ahead and wrote the R group, the side group of each of these amino acids. Your instructor may want you to memorize all 20 different side groups. When I was taking biochemistry as an undergraduate, my instructor expected us to memorize all 20. When I was teaching this biochemistry, introductory biochemistry, I never asked my students to know all 20. So you'll want to clarify with your instructor whether or not you need to memorize all 20 of those different amino acid side groups. If you don't need to memorize them, just make sure that you know where to look them up. You can do it easily in your textbook, I'm sure, but you can also do it quickly with a Google search. All right, so these are the side groups for these 4 different amino acids for problem number 4. All right, so we're going to write either on the outside or on the inside. Where are they going to be found? On the outside, oriented toward the aqueous environment, or on the inside, coagulating together away from the aqueous environment? Well, leucine is composed of all carbon-carbon and carbon-hydrogen bonds. So this is a nonpolar neutral side group. And remember that that makes it hydrophobic. Nonpolar side chains are hydrophobic. So we would expect to find leucine on the inside of a globular protein oriented away from the aqueous environment outside of the protein in the body, the aqueous environment of the body. Okay, glutamic acid is CH2 with carboxylic acid at the end. Remember that at physiological pH, these are typically deprotonated. So really it's going to look like CH2, CH2, CO, O-, in which case we would more accurately call it glutamate for the negative charge here. And this is going to be a very hydrophilic side group. It's charged, and that charged group can be attracted to the partial positive, partial negative charges of a water molecule or some other partial positive, partial negative charged molecule. So this would be found on the outside of a globular protein where it could interact with the aqueous environment of the body. Aspartic acid, same thing going on here. It's going to be charged at physiological pH. which means it's more correctly referred to as aspartate. Okay, and that charged side group is going to be hydrophilic, interacts with the aqueous environment, so it's going to be oriented most likely on the outside of a globular protein where it can interact with the aqueous environment of the body. All right, last but not least, we have valine, all carbon-carbon and carbon-hydrogen bonds. This is a non-polar neutral side group. Which means that it is hydrophobic, and the hydrophobic side groups will orient toward the inside of a globular protein away from the aqueous environment. So most likely we would expect to find this on the inside of a globular protein. Okay. Number 5. What levels of protein structure can be affected by denaturation? Okay, so remember that the levels of protein structure include the primary, the secondary, tertiary, and quaternary structure. And it's the secondary, tertiary, and quaternary structure that can be affected by denaturation. So let's write that here. The secondary, don't write primary, the secondary, tertiary, and quaternary. Secondary, tertiary, and quaternary structures. Remember that the primary structure is not affected by denaturation, that the primary structure can only be broken apart by the chemical reaction known as hydrolysis. So that's what would affect the primary structure. Denaturation affects just the secondary, tertiary, and quaternary structure. All right, last but not least, number 6. What level of protein structure is determined by, and we have an A through C here. I think we can fit it on the board. Okay, so A, what level of protein structure is determined by peptide bonding between amino acids? That's describing the primary structure. So for A, you should write the primary structure. Okay. For B, what level of protein structure is determined by non-covalent interactions and covalent disulfide bonds involving the amino acid side groups? So 2 levels of protein structure are stabilized by these types of non-covalent and covalent disulfide bonding. And that would be the tertiary and the quaternary structure. So I'm going to write here the tertiary and the quaternary structures. Remember that they are stabilized by the same types of interactions. And part C, what level of protein structure is determined by hydrogen bonding between the oxygen off of the carbonyl of a peptide backbone chain to hydrogen atoms off of nitrogen atoms in the peptide backbone chain. So now we're talking about no side groups being involved here, just carbonyls of the peptide backbone chain being hydrogen bonding to NHs also off of the peptide backbone chain. Those are interactions that stabilize the secondary structure of a protein. So part C, you should write secondary. Okay, and that will conclude our fairly short problem set number 23 I hope that you will consider tuning in to tutorial number 24. Thank you very much.